All right, we're going to get started here in a minute. So please come on in, sit down. Got some people trickling in. This is free flow, so people can come in and go out. All right, so for those of you who don't know me, my name is Garth Spellman. I'm the one who's giving the talk today. I am curator of ornithology in the Department of Zoology. I use he, him pronouns, and I really appreciate all of you coming today. So I'm going to talk about today some work we've been doing for the last 5 years in my lab, mainly in collaboration with John Peacock, who's listed in the front there, who was a postdoc at the University of Colorado Anschutz in an otolaryngology lab there. And a lot of what I'm going to talk about today is a paper that was published this summer in the Anatomical Record. So if you want to go and read a nice monograph about bird ears, you should go do that. But you don't have to after this, because you're going to know it all. Okay? So it's all okay. But before I get going into the research, I also want to talk about inspiration for why I'm really interested in this work. So there was a guest essay by David George Haskell, who's an ecologist at the University of the South in Tennessee. And this was published this summer, and I thought he captured sort of my feelings about this research and why it's important to me. He wrote an essay about working in his garden that he does every year. And he loves to work in his garden in spring because he can chart bird migration through sound. He listens to the birds around him while he's planting his trees. And this last spring, he asked his wife about his favorite bird. He's like, it's been 10 days late. He loves blackpoll warblers. And he says, they haven't arrived yet. And it's just shocking to me because we've had such a warm spring. And she pointed out to him that they moved through 2 weeks ago. And the reason why he didn't notice it is because he lost his ability to hear those high-pitched songs of warblers. And so his worldview sort of compressed because of that. Something he loved was lost to him because he was not able to hear. And that is actually happening to somebody who's dear to my heart in my family, my father. He had the same experience about a decade ago. He loves to feed birds in his backyard, and he watches the warblers, and he lost his ability to hear yellow warblers that nest in his backyard in Fargo, North Dakota, about a decade ago. And that has only progressed. He's needed to have additional hearing aids and other things. And so it makes me think about, how we view the world and how we view things around us and experience them. So to me, it's sort of a personal journey as well. And so we need to talk about birds and hearing. And in answer to Paula's question, does a bird, hear a seed if it drops in a forest, if there's no one there around to hear it? In the email she sent out, yes, because birds have ears. Okay? For those of you who didn't know that, birds have ears. All right? They do. They don't look like this, okay? So they don't have these crazy pinnae that stick out of their heads, because that wouldn't be very aerodynamic, right? So they have evolved to look quite differently. And it's easiest to see in some other birds. So this is a long-eared owl. Are those ears? Those feathers? Who says yes? Okay, no one does. Thank you. Okay, long-eared owl. It's a misnomer. Those aren't actually ears. Those are just feathers coming off of its head. Where are its ears? Right? What direction do they face? Do they face forward? Do they face to the side? So in an owl's, they're facing forward. And those discs around their eyes actually are modified to sort of help direct sound as well. Right? It's a lot easier to see in birds that have no feathers on their heads, okay? So a cassowary, you can see exactly where the ear hole is, and then also our local turkey vultures. You see that little opening down on the bottom of its head. So yes, birds, in fact, do have ears. Other birds that are fully feathered have modified feathers on their heads, so they're called auriculars, and they're different in composition than the other feathers on their head, okay? They oftentimes are not as pennaceous or well-veined, so it allows sound to penetrate through. And one of the most amazing things that you can often see in birds, especially their feathers, oftentimes uniquely colored feather patches are disconnected from other plumage feather patches in their genome. So the genes that control coloration on those can be turned on and off at different times and can be controlled differently, and therefore you can get different colors that appear on those feather patches. The auriculars are one in particular where we've identified the genes that are responsible for coloration in auriculars, and we can sort of go back in time to see when that unique coloration occurs. But you see this in a lot of birds. Those auricular feathers that cover the ears are a different color. All right, so now that we've clarified that birds have ears, what do we know about birds and hearing? Because most bird research on sound actually gets done by the producer of that sound and not the receiver of that sound. So we don't know a lot about it. So this beautiful little guy is a Costas hummingbird. And about 10 years ago, some researchers went out to study Costas hummingbirds and find out how they hear, because they produce some of the highest frequency sounds, so would be considered ultrasound, of any bird. We can't actually hear that frequency, so we can't hear the sounds that this hummingbird makes. We don't understand what its soundscape is. And so they went out to study this, and they captured Costas hummingbirds. They anesthetized them. They stuck probes in their heads to get an audiogram to figure out what the birds hear. And they said, well, the birds, they have no brain sensitivity at the high frequency sounds. So they said they must be producing sounds they don't hear. It came out after they published the study that they actually used an anesthetizing agent that dampens the brain's ability to, right, to sensitivity to sound. So it's not easy to study these things. Now they're studying it more from a receiver end in the natural ecology. So they produce the sounds that Costas are making, play it back to them, and see if the birds respond. And they've found in several hummingbird species now that, in fact, they respond very sensitively to high-frequency sounds. Shocker, right? I mean, birds are producing these sounds both vocally and with modified feathers when they're aerial displays. It's not a shock that the receiver, so in those displays, can also sense those sounds. It's not surprising. We also know very little about sound on the other end of the frequency scale for birds. So this would be infrasound. So very, very deep sounds, long frequency waves. We don't know really if birds can hear them, but it's been thought for several decades now that birds use infrasound to actually navigate during migration. We just don't know how they're doing it. And recently, a hypothesis has been put forward because lots of long-distance flying seabirds are thought to navigate via infrasound, especially in polar oceans. You think about South Polar Oceans and all the islands that sort of surround Antarctica. So there are waves that are constantly hitting the shorelines, and those waves produce these very low frequency sounds that travel great distances across the South Polar Ocean. And it's thought that these birds are able to detect those sounds hitting different shorelines, and that is how they find their way back to their nests, how they find their way back to their young when they fly far away and they can't see the islands. They are using infrasound to navigate. And yet, we don't really understand how the ear functions. And so we started about 6 years ago trying to get an idea. And granted, I'm an ornithologist who studies evolutionary biology and evolutionary genetics, so I had no idea how to enter into this world. And this is the anatomy of a bird ear. So we chose to focus on one area, and that is the middle ear. So this is the tympanic membrane that's right there. The middle ear is the area between that and the cochlea, which is right here, which is called your inner ear. And there's reasons why we decided to focus on that. The cochlea has hair cells, right, that are detecting changes in water pressure, or fluid pressure that occur in the inner ear, and it's translating that into electrical signals that are sent to your brain to interpret sound. I'm not a cell biologist. I'm also not a neurobiologist, so that was really hard for us to do. And I also work at a museum, and we don't have lots of living things, so we don't have live cells. One thing we do have a lot of at a museum is a freezer full of cadavers, right, that are being prepared, that will be prepared for our collections. Fortunately, most middle ear work that has been done on humans and mammals that are models for humans is done on cadavers, because the mechanism, that middle ear mechanism, is a mechanical mechanism and can be studied in freshly dead animals. And so we learn a lot about the mechanics of that by studying those animals. So we have access to that. And so the first thing we did was we wanted to study how sound waves traveling through air are transferred from air pressure changes along the tympanic membrane to mechanical energy differences along that area and then into the inner ear. So it's called the mechanics of that middle ear, how it vibrates in response to sound and exposure to sound. And we did this in cadavers at CU Anschutz. And so right in the middle of the pandemic, we published our first papers on this. And so what we did was we took birds. This is actually the outside of a bird ear right here. And we put a little tiny speaker on the inside of the bird ear. And then we also have a probe microphone to make sure that the sounds that we are exposing the eardrum to, right, are the frequency that we want them to be and at the decibel level that we want them to be. And then we go in and we dissect out the inner ear so that we can put a laser vibrometer on the back side of the middle ear to look at how it's vibrating in response to exposure to different sounds. Does that make sense to everyone? All right, good. So we do that, and then you get something that results like this. And so all of you know what a peafowl is, right? Peacock, peahen, combine them together, that's peafowl. Okay? So mallard duck, wood duck, and then a merganser in this window. And so what this shows is that there is a peak frequency. So this is your frequency in hertz. So that is going to be the sound wave, right, that goes across there. And so you get a peak energy transfer. That just means that mechanism in the middle ear is vibrating at its peak frequency at a certain, right, sound level. And so we can get these curves for all birds. And we actually find something very similar to the few audiograms that have been produced by birds, is that these curves actually capture almost perfectly sort of brain sensitivity to these sounds. It's not surprising. The bird ear is adapted, right, to hear levels that the mechanism is working at. So if the mechanical middle ear is not transferring a sound frequency, then the brain is not going to be sensitive to that. There's no sense in that. So you have limitations based on this mechanical mechanism that is in the middle ear of a bird. And so that's what these curves represent. And it shifts a little bit depending on the bird. And a lot of it has to do with the size of the animal. So a hummingbird would be shifted towards the high frequency sounds if we had a hummingbird on here. The probe's too big to get into a hummingbird ear. We can't actually do anything with hummingbird ears. They're just way too small. So, but we do have, I think our smallest that we've done this for is a bush tit. That's a bird that's about 4 grams, 4 to 5 grams. And so it shifts, as you would expect, into the high frequency range. So small mechanisms transfer those high frequency sounds a lot easier. And then the big birds are shifted more towards, right, the bass. So they hear those low frequency sounds better. And that's what this shows right here. This is looking at the bone, the columella, that is in the bird's middle ear. And as they grow in size or get longer, they are transferring low frequency sounds much better than high frequency sounds. So the nice thing about this work is it allowed us to model how birds are hearing, and it also allowed us to predict what anatomical features of the bird's middle ear are responsible for most of the variation in the frequency of sounds that they hear. And what we found is that the bone in the bird's middle ear, birds have a single ossicle, a single bone in their middle ear. Mammals have 3. Right? That variation in this single bone explains about 70% of the variation in sound that is transferred across that middle ear. So we started thinking, oh wow, we can study these bones and learn a lot about how birds hear, and we can figure out whether or not evolution is having an impact on the adaptation of their ears, just by studying variation of this bone. And so that was what we started doing during the pandemic, when we could get in here and start looking at bones, because we couldn't get out in the field to actually get audiograms from birds. And so we shifted our focus to studying these bones. And that's where we are today. And so we actually had a number of goals for this. We want to know, is this diversity of form adaptive in birds? And so we started describing variation in the columellae of birds, the bird middle ear bones, in the light of modern day phylogeny. So that just means the evolutionary tree of birds has changed a lot in the last 100 years as we know more about it through DNA. And now we can look in modern trees whether or not we see any particular forms in these middle ear bones that could be explained by that evolutionary tree. And so there were previous columella studies, but not by many people. So first was back in 1901, a German scientist, Krause. And where's Frank? He would appreciate this. Krause. Oh, thank you. Thank you. I didn't say it correctly. He published his monograph on avian columellae back in 1901, and it's in German, and I had to use Google Translate in order to figure out what it said. So I should have just given it to Frank and had him read it to me in English. No, he probably wouldn't have appreciated that. But he did describe variation in the columellae in about 70 species back in the day, but didn't know anything about really the evolutionary history of birds, and he just described the form and made drawings, which are actually really quite nice, of the columellae. The only other person to really focus on bird middle ear bones was Fiducia, who is sort of an avian paleontologist. And he did most of his work back in the 1970s. And he originally described what he considered to be an ancestral form. Like the first birds would have had a columellae that looked like this from a kiwi. That's Apteryx right there. And so that's what he said. And he also described diagnostic forms of various clades. So the upupiforms, that's a hoopoe. Does anyone know what a hoopoe looks like? They're really cool birds. So kingfishers, so bee eaters, motmots, and toadies. So, okay. And then he also made several erroneous conclusions. He thought that the shoebill, which I pictured earlier with ears, That's a shoebill right here. It doesn't have ears like that. But he actually said, well, the structure of that bone suggests that the shoebill is a stork. But we know through DNA that, in fact, it's not a stork. So it's a weird-looking pelican. So he got some things wrong based on studying these bones. And so we kind of wanted to go through and correct a bunch of what he was saying. So what did we do? We went in and we got bones from 401 species. So we got about 120 from here at the Denver Museum of Nature and Science. And then we went to the skeletal collection at the Field Museum of Natural History and got the rest of the samples that we needed. And we got 40 orders and 146 family-level clades. This is what some of the skulls look like. And it's not very glamorous work. You take a little forceps and you dig around in the ear canal and you pull out the bone. And so it's pretty tedious, and you're looking under a microscope for many hours. And then we took measurements and described sort of how these bones looked across the groups. And so in relation to the ear, this is the inner ear right here. This is the tympanic membrane. And so we're focusing only on this bone that is right there, that acts as sort of the piston in that mechanical apparatus. And so what we do? Well, we made 9 measurements, and I'm not going to go into details about what those measurements are, but I'm just going to tell you about what we found at the end. So we have a number of results that we weren't so surprised with. We did see that size, right, scales accordingly across these bones. So essentially, your foot plate at the end gets wider as the bone gets longer. That's the scaling, right? It's scaling. As birds get bigger and this apparatus gets bigger, these things have a tendency to scale together just fine. We also found 3 different forms. And the only thing you really need to know about this is your sheet forms are only found in small birds. So small songbirds, generally, they don't have a rod-like shaft that comes off of that foot plate. It has a tendency to be a sheet. And that has functional significance, and we're trying to figure out what that is right now. Other things, as the foot plate area and the length, they scale together. And then we also found some weird things, that the basal area is generally, right? It's generally small compared to the foot plate area, but you have some other patterns. And you're all looking at me like, okay, what? Right? But we're going to get into what this all means. All right, so if you have a basal area that's the same size as your foot plate area, your columellae look like this. They're hollow, and they have this very wide basal area and the foot plate that goes into the inner ear. And we believe that this is important ecologically. We don't know why yet, but we're going to get there. So the other thing, we get to fiducia. He thought kiwi was a primitive form. And so before we talk about whether or not it's true, I thought this would be a good time to describe to you how to prepare a kiwi. Okay? So first thing, got to get your kiwi. Okay? You got to get a knife and a razor. Razors really help. So then you lop off the legs and the bill and the head and the legs, and then you shave it. Okay? So because the skin, that's actually where a lot of the good nutrients are. So you need to save that. Those of you who eat kiwi, so make sure you save the skin, and you chop it up, and then you can eat it. All right? Okay. That's how you prepare a kiwi. Andy can attest to that, right? All right. Excellent. Okay. So fiducia thought that the kiwi actually had a primitive form. And if you actually look at ostriches, rheas, tinamous, and cassowaries, they have very, very different forms of this middle ear bone. So kiwi wasn't primitive, but if you look at all the geese and the ducks, they look exactly like a kiwi. That means a kiwi's ear, right, has essentially converged upon the form that you find in a completely different group of birds. We don't know exactly why yet, but we're looking into it. Other things we found is that souliforms, so your frigate birds, your boobies, your gannets, they all share a derived morphology where they have these very pronounced angled columellae, and that's not true. That's not true with the anhinga here, which has a very different form. And we believe this actually has to do with diving behavior. And we'll talk about that a little bit later. And then the other thing, the only other real thing that we found was we found a distal fenestra within a certain group of raptors, so that go to hawks and eagles, and they define this group of birds, the Aegypinae, Aquilinae, and the Butyonidae. So those are all, right, raptors. And it's exclusive to Old World. It's separate from Old World vultures. And this is one of the few defining characteristics that actually identifies this clade that was identified by DNA. So from an evolutionary standpoint, this was pretty cool. But for you, it's probably boring. So what about functional significance? And that's what we're getting into now that we've published these morphological studies. Now we can start to get into hypotheses of why the bones are shaped the way they are. And back in 2006, Mills and Zhang, they looked at the middle ear bone of gannets. That's this diving bird right here. And they're plunge divers. So they go at great speeds and hit the water. And, oh, it's too bad. Actually, that's not their columella. It's not on the slide. I don't know where it went. But their columella is really angled, and it's almost ribbon-like. And that is thought to actually be like a spring, so that when they hit the water, the static pressure loads that they're exposed to when their head hits the water doesn't damage the inner ear. And so it acts like a spring and doesn't put that pressure into the inner ear. And if you look at it, this is weird. It looks fine on my screen. So we also find that in two other groups of plunging divers. So kingfishers, how many of you have seen belted kingfishers in Colorado? So when they also plunge dive very similarly to gannets. And their middle ear bones are hooked. They have a pronounced hook to it. And the same with the only plunge diving songbird that's out there, the American dipper, which you find around fast-flowing streams in Colorado. It dives under the water looking for aquatic insects and invertebrates. So you actually see this adaptive trait appearing multiple times in multiple plunge diving birds across the tree of life. And so we think we have a lot of other characteristics that we can look at in the morphology of this bone that'll tell us something about the adaptive pressures that these birds have experienced as they've evolved their hearing and their feeding ecologies as we move forward, which is really cool for somebody who studies adaptive variation in birds. But the work also actually has a translational application to it. So if you look at middle ear implants that are put in human ears that are damaged through exposure to very loud sound, so you can think of soldiers that are on the battleground and bombs are going off close to them, or they're exposed to blast sound. Oftentimes they damage the mechanics of their middle ear. So the three bones that are found in humans can break, can snap, and that ruins your ability to hear. And so they put middle ear implants in victims of blast damage, and they're called ossicular replacement prosthetics. Some are partial, others are total. But they are modeled after a single bone model, a single ossicle model, and not a three bone model, because that would be really difficult to replicate. And So we actually believe that the work we're doing by looking at the form of the columella and seeing how it transfers sound can help us design better middle ear implants for victims of middle ear damage. And so we actually started conversations to work with a prosthetic designer out at CU Anschutz to see if this was even a possibility. And we think it has potential as we move forward. And so that's our middle ear work at this point. That's what we've done. We are going to continue to do this work in birds as much as we can. And I hope you learned a little bit, at least how to skin a kiwi, and that you know more about bird ears. So thank you very much, and I'd love to take questions. Jenna has one.
I have a question. So when the lead author's name is Peacock, is he obligated to study birds?
You know, he didn't study birds at the beginning. It wasn't till we talked. And he originally came because he was interested in variation in mammal ears, because the work he was doing at Anschutz was mainly on chinchillas and naked mole rats, which are actually used as models for human hearing. Chinchilla ears are actually, if you look at the frequency range that the ears transfer, it's almost perfect for human models. So,
Question over here.
So I know some cetaceans, so like dolphins and whales and stuff, have a self-deafening mechanism because they emit these really loud sounds. So they have like cartilage in between their actual ear and their skull, so they don't deafen themselves. Do any birds have a similar mechanism?
So birds in their extracolumella, which is actually the soft tissue part that attaches to the bone in the middle ear, there is a single muscle that is associated with that mechanism. And the function of that muscle, there's very little known about it, but it does vary in size across the avian tree of life. And so it could potentially serve some sort of function like that, but we don't really know, just because nobody's paid any attention to it. Mm-hmm.
I have a question. Do we know how increases in incident sound in the environments are affecting bird hearing?
So we don't know how it's affecting bird hearing, but we do know how it's affecting vocalization. So Paul is kind of referring to, if you have lots of background noise, how is that affecting birds? Well, in birds, they change their frequency of their songs in relation to the background noise. So many urban birds, for example, the frequency of their songs in urban environments gets higher than birds that are in rural environments because of the low-level, low-frequency background noise. So it's a way for them to distinguish, get their song to distinguish and carry over that background sound. And it's always within a range that they can hear. We know that. And so I don't think that is going to change very much. Mm-hmm.
Duck, Megan.
Is there any kind of hint at a correspondence between the structure of the syrinx and these ear bones?
That we also don't know, if there's a correspondence with the structure of the syrinx. And it's kind of like that hummingbird story where, why would they produce sounds that they can't hear when they originally studied it? And the syrinx is limited in the types of sounds it can produce based on the structure of the syrinx. I don't know. I imagine that those sound frequency spectrums, they evolve pretty closely in tandem with one another. Because the production of sound that an organism couldn't hear is not going to have, right, any evolutionary consequence, essentially.
Thanks. Based on your sample, would you be able to reconstruct an ancestral condition of this bone and any like sound inferences in that ancestral bird?
We absolutely could. John Peacock is actually working with Tyler in some paleo animals to look at the columella as well, to look at the sound spectrum, or soundscape for those animals in the past. But the models actually are really quite amenable to prediction. We still haven't done the full phylogenetic reconstruction of what the true ancestral state might have looked like. That's part of the sort of next phase of the work.
Other questions? I don't throw to brand new directors.
Hey, great talk, Garth. That's awesome. I have so many questions, but I'll catch you on many of them later. But so 2 questions. One's maybe a behavioral vocalization question. But do you know when during ontogeny, do parental birds actually demonstrate a frequency shift when they're teaching vocalization to young birds?
I believe it's been demonstrated in some species that they do, but it's not universal.
Yeah.
So across the board.
Yeah, I think it might be tough data to have, but just in terms of, because you're showing these nice kind of structural relationships which have implications for frequency, receptivity, or selectivity.
Yeah.
I just wonder if you could look into songbirds or specific groups of songbirds. That might be a neat thing. But then I guess another one, what about, do you know anything about any of the kind of structural mineralization kind of parameters of just kind of generally across relative mineralization, I guess?
Of bones, yes.
Of columella.
So that's one thing that we've looked at least when we were at the Field Museum. We got some series of different aged birds. So they had a lot that were fledglings from adult, and it looks like ossification of this bone occurs very early within the birds. So based on those small sample sizes. And that it doesn't vary in size that much from adults. Okay. One of the things we're looking at in relation to the columellae that are highly angled, so in diving birds, that we need to get an idea of flexion in that bone to see whether or not even the bones that are curved, if they're really rigid, then they're probably not bending under pressure loads. That's a really hard thing to get to because it's really hard to get these bones. We don't want to expose them to pressure so that we break them. Sure. And a dried bone is not going to get you much of an idea of what a living bone, right, looks like. But one thing we've looked at in the new microscope we have downstairs, that Keyence microscope, we can get an idea of bone cell density across different types of columellae. And so that would be essentially counting the number of cells that you have. And bone scientists, people that study bones, know that flexion is related to cell density as well. And so we can go in and we can count the spaces between the cells using a microscope along the columellae and compare across animals to see if there's any relationship to how possibly flexible the bone will be across clades. If you want to do that work with me, that would be great because it's really boring and tedious.
One last question. Where's the pelican?
The pelican. Oh man, it's not here. I do have a pelican. And it is a weird looking bone.
They tend to be that way in many parts of their skeleton, that's why I
Exactly. This is the American white pelican. So it has the longest columella. It's got lots of fenestra through it, and it's just crazy in comparison to others. It's an outlier when you look at the mechanics of it. So it doesn't transfer sound very well. They can still hear, but it's not as good as most birds. And so they're weird. There's one group of birds that has an even longer middle ear bone. And we don't have them yet, but the largest of the albatrosses, like royal albatross and wandering albatross, have a much, much larger one than you would expect from the size of the bird. And we wonder if that has to do with infrasound adaptation. Mm-hmm.
If you go back to the last slide you have of all the bones, I was wondering, do you know the reasons for some of the bones to have the holes in them? Like, not very many have them, but I think like 6 and 7, and then the weirdest one I noticed was number 34. And also, what bird is 34 from?
So 34 is the laughing kookaburra. So it's a kingfisher, and it's got a real hollow end to it. So if you look at the back side of that, so it's got that big hole, and then the back side is solid. And so it's kind of a sheet like that. And most of the kingfishers have a very similar form, and then the very end of it is really curved like you see in 35. This is a small kingfisher species, but yeah, like 31, 30, 32, that all have those big holes with sheaths around them. Those are all kingfisher kind of morphologies. So you see them in bee eaters too, which are kind of closely related. But you're right. They're really weird looking, and we don't know exactly what they're doing yet. So
this might be a dumb question, but have they ever seen these bones in the fossils of the lineage that arose, that gave rise to birds?
So there are very few columellae that have been preserved in the fossil record, but Sue actually has one in her skull. So, and it's really long, as you would imagine. She was not hearing very well, so it probably, I think it's about that wide, and at least the parts that are preserved are about that long. So you think of the sound frequencies that a T-Rex was able to hear, it probably was very, very low frequency sound. I imagine she had her ear to the ground a lot.
Other questions? Let's thank Garth for a great lecture.
Thank you.